Good afternoon. This is Group 4 and we are talking about H1N1. I'm Rejoice Esnam Yaku, IDPB PhD student.
Okay.
I am Bulbul Ahmed. I am IBS PhD student in the group D, and registration number is ID number 0972087.
And this is Sajal Moshel Shajib from Masters in Biology program.
So in 2009, a new variant of H1N1 was detected, and it started before then. So it is known that the H1N1, which is also called a Spanish flu, started in 1918 and then continued throughout the world, causing the deaths of over 10 millions of people worldwide. So the CDC named it as a pandemic because it was worldwide. So therefore, elderly persons over 60 years are known to be immune to H1N1, probably due to a previous H1N1 infection. And in 2009, a new strain of the H1N1, which is called H1N1 PDM09 virus, was found and is commonly known to target the lower respiratory tract, especially the distal lung cells. Human alveolar epithelial cells are a key part of the lung defenses against respiratory pathogens and the primary target for pandemic and severe influenza. So the H1N1 is a type of influenza A virus. It's an automyxo virus, a single-sense negative strand RNA. And influenza A viruses are classified into different subtypes. And there are two proteins that are found on their surface. That's the heme agglutinin, that's the H, and then neuraminidase, which is the N.
The H1N1, that means the swine flu virus, it first creates the it creates both cell-mediated and humoral immune response. So it involves MH1 and MH2 both in this case, as well as the TH1 and TH2. The first acute response happens in the mucus cell, in the mucus of our nose and air passage. That happens by innate immune response. First, it activates the humoral immune response that is characterized by an early rise of antigen-specific immunoglobulin, followed by affinity maturation, isotype switching, and ensuing rise in antigen-specific immunoglobulin A, immunoglobulin G, and immunoglobulin E antibodies. However, influenza A viruses are contagious pathogens responsible for severe respiratory infections in human and animal worldwide. Upon detection of influenza A virus, that is, we are talking about influenza A virus because H1N1 is one kind of influenza A virus, and that is that infects the host immune system. And the host immune system aims to defend against the H1N1 and clear the virus infection. Innate immune system is comprised of physical barriers that includes mucus and collections, various phagocytic cells, group of cytokines, interferons, and interferon N stimulated genes, which provide the first line defense against the influenza A infections. However, later on it activates the adaptive immune response that is mediated by B cell and T cell and characterized with antigen specific memory cell capturing and neutralizing the pathogen. The humoral immune response functions through the hemagglutinin specific circulating antibodies to neutralize the H1N1.
There are two types of vaccines that are currently used. Trivalent inactivated vaccines, TIV, elicits humoral immune response, and live attenuated influenza vaccine, LAIV, elicits mucosal protective immune response without causing clinical diseases.